Okay, I wanted to go over again two gene crosses, which I didn't quite finish in last class. So we were talking about a parent generation that had round pea plants and purple flowers, and they were crossed with a pea plant that had wrinkled peas and white flowers. We mentioned, first of all, that the F1 would look round or have round peas and purple flowers. So since we see both of those characteristics in the offspring, we know round is the dominant phenotype and purple is the dominant phenotype. In the parent generation, they're both true breeding, so they were homozygous. Since round peas are dominant, we'll use a capital R to represent the allele for the dominant trait. So round peas, the genotype for this plant is big R, big R. The wrinkle is recessive, so little r, little r. And then purple is carried on a different gene on a different chromosome. Since purple is dominant, we'll use P for that. So since this is a true breeding plant, it would be big P, or capital P, capital P. And the white flowers would be lowercase p, lowercase p. And I like to use a Q-looking P to highlight the difference between the uppercase and the lowercase. When these are crossed, this parent can only make one type of gamete. During meiosis, these two genes separate, and only one of each gene is passed on to the offspring. So the only gamete that this could make is a big R and a big or capital P. And this one, the only gamete that could be made is a little r, little p. So if we draw a little punnet for these, the F1s are going to be heterozygous. Now Mendel went and allowed these to self, allowed them to fertilize themselves and reproduce. And so to see the outcome of the F2, we have to figure out all the gametes from each of these parents. So each of these genes are on a separate chromosome. So you'll get one of each of these alleles passed on to a gamete. So you can have big R, capital P, big R, lowercase p. By the way, I mentioned this yesterday, but I like to actually do this in a consistent pattern where I make the gametes in a consistent way so that I can find them then on the Punnett square quickly. If I randomly order things, like if I switch the letters, then it makes it difficult for me to figure that out. And by convention, we usually start with the capital letters, but we don't switch. We use capital letters when we are making actually a zygote. We don't switch when we're writing the gametes. We keep it in the same order of genes that there were before. Then we have lowercase r, uppercase p, and lowercase r, lowercase p. So this one right here, this parent produces these gametes. This parent also will produce the same gametes because it has the same genotype. So then we draw a Punnett square to show how this works. And this parent, we use all the gametes that it produces. Okay, and then the other parent too produces the same gametes. So I'm going to write those down too. Okay, and then I fill out the combination. Just be aware again, there's always one of each gene in each of the gametes. And then the zygote is going to have two alleles for each gene So this is big R, big R, big P, big P. Okay, big R, capital P, little p. And I'm going to go through and do the rest very quickly. Okay, and then you have to figure out the phenotypes. Oh, I'm kind of blocking. Okay, so if they have a capital R and a capital P, they are going to show round and purple. If they have capital R, for instance, this one, and two lowercase p's, they are going to be round seeds and white flowers. Okay. Then if they have two lowercase r's and a capital P, then they're going to have wrinkled seeds and purple flowers. And if they have both the recessive, homozygous recessive for both the gene of R and P, they are going to be wrinkled and have white flowers. Okay, so there are only these four possible phenotypes that can be produced. And to figure out all the ones in the correct ratio, what I like to do is mark them in the box so I know which ones I've done before. So for the round and the purple, I'm going to use a little red dots. Okay, so that one's round and purple, 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 round and purple. Okay, if I count those 4, 5, 6, 7, 8, 9, round and purple. Okay, for the round and white ones, put a little triangle in the corner. This is a round and white. This is a round and white. And this is a round and white. There are 3. Okay, and then for the wrinkled and purple, I'm going to put a little circle, blue circle. So this is a wrinkled and purple. This is a wrinkled and purple and a wrinkled and purple. There are 3 wrinkled and purples. And the last one, wrinkled and white, there's only one of those. So the phenotypic ratio here is a 9 round and purple to 3 round and white, 3 wrinkled and purple to 1 wrinkled and white. On all my questions, I will ask for genotypic and phenotypic ratios. You always have to include what we are talking about with genotypic and phenotypic ratios. I can't just say there's a 9:3:1 ratio. I have to say what it actually is for each thing. Okay. By the way, this is a ratio that you should memorize. If you have 2 dihybrids, they're called, 2 organisms that are heterozygous for each gene, then you're always going to get a 9:3:1 ratio, as long as the inheritance follows this simple dominance inheritance. We're going to talk about some where they will not have a 9:3:1 ratio, but usually this can help you get started in figuring out what the ratio is. Okay, I'm going to stop there. Have a great weekend, guys.